cartographer method developed by US scientists. And the result, quantitative character, it should have a continuous segregation pattern. And for stem swelling tests, we found more or less segregation continuous, that is, they are according to their stem swelling size, they are continuous. And also here we found stem swelling types, we measure more or less continuous among the BC1 population. Then using BC1 population, we developed 9 linkage groups. We know BC1, kohlrabi is a basic oleracea. It has 9 chromosomes, and to align this, we found 9 linkage groups. And after software run in QTL cartographer, we found 3 QTL in chromosome 2, 3, and chromosome 5. Then this QTL we detected in BC1. Then after that, generally all QTLs are not significant. Sometimes false QTL we found. So to confirm this QTL, then we went BC1 F2 population. BC1 F2 population of this line. Then we used BC1 F2 population that is heterozygous for QTL1 position, but homozygous for other QTL position. Then we found very good segregation in this line, that is BC1 68F2. And this for QTL1 heterozygous, they producing very good segregation, also their distribution curve. And we then measured, we then mapped a QTL1 in chromosome 2. This is a very precise map with a significant near about 14 logarithm score. Then we also confirmed the QTL3 in BC1 F2 population, that is BC1 F2 57. BC1 F2 57 also produced very good segregation in their stem swelling phenotype. And we found a unique QTL in chromosome 5 of Basita oleracea. So, discussion, stem swelling of kohlrabi is important for kohlrabi, and not only for kohlrabi, it is also for brassica and other stem swelling producing plants. We identified 3 QTLs by using BC1 population, and then we confirmed the QTL in BC1 F2 population. And the candidate marker of this QTL we found in QTL1, this gene is producing, that is alignment with Arabidopsis thaliana. And in Arabidopsis thaliana genes, they are responsible for ATP binding, ATPase, that is energy production mechanism. And another QTL candidate marker, it contains in ARF, that is auxin response factor. And auxin is important for plant growth and development. So we are thinking that according to alignment of orthologous gene Arabidopsis thaliana, our marker is authentic. And so we want to confirm this QTL by position mapping and cloning to find out the more precise and unique responsible gene for stem swelling tests. That is all about my presentation. Maybe.
Yeah, thank you for the talk in time. And now the session is open for the discussion. So if you have any, yeah, please.
So normally, quantitative phenotype is controlled by dozens to thousands of genes. Normally. So did you find how many potential genes actually responsible for, this type of, phenotype is stem swelling?
Yeah, this is. Here position, very long position, many genes in this one. Then we found.
Did you count actually how many potentials?
in QTL1 position, we found near about 150 genes, but we don't know which gene is. We should confirm by position cloning.
Okay.
Any other? If not the case, let me conclude this by thanking him. And now, the next invited talk is by Mr. Muhammad Ibrahim Hussain. He is also from Niigata University, and he is going to talk on the topic activated glial cells in the brainstem of Dystonian mutant mice.
Assalamu alaikum wa rahmatullah. Today I will like to talk about a part of my research which is the title is Activated Glial Cells in the Brainstem of Dystonian Mutant Mice, and I am from Niigata University. So in my lab I mainly works with a project called dystonia. So the first question comes, what is dystonia? So you can see by simple term, dystonia is a movement disorder. So in this type of disorder, person can move because when they try to move, we know that when we try to move or do something, our muscle works. But in this type of patient, their muscle, antagonist and agonist muscle together, so that they cannot move their limbs or legs or something which is affected. So it can affect the you can see it can affect the hands and also the leg and also the writing ability. So what is the cause of dystonia? So far there is no exact cause is known about dystonia, so our lab is looking for it, but so far about known that dystonia is mainly occur due to the abnormality in a region of the brain called basal ganglia. So it actually the region where all the signals are coming for the movement of the muscles and also the sensory movement from muscle to the brain. So this is the primary cause where the abnormality can occur, and it's a very rare disease actually. But it can also inherited by generation to generation, and it can also occur by some drug or birth injuries, strokes, or as some neurological disorders as well. So in our laboratory, we actually generate a mutant mice by using gene trap technology. So in this technology, this is the Dystonian locus, the gene which is responsible for the mutant Dystonian gene is here. So we inject LAG-G, which is a beta lactase. So by using PCR, we can easily detect the dystonin and also the homozygote and also the wild type as well. And I like to mention that our mouse phenotype is actually similar to that of the human phenotype as well. So by using this mouse model in 2014 from our laboratory, the first paper is published. So as I mentioned earlier, that in wild-type mouse, you can see the triceps and biceps muscle, which movement our forelimb. You can see those are not working in the same time. But in case of our homozygote mouse, which is the mutant mouse, so you can see it is by dotted line. You can see the muscles are contracting at the same time, so they cannot move their muscle. And in this paper, in the spinal cord, you can see there are lots of abnormality, which is indicated by arrowhead and also arrow. And you can see this is called DRG, that means dorsal root ganglia. It is the place where the PNS and the CNS is connected to each other. And we found that in DRG, that means dorsal root ganglia, it is quite smaller than the wild type, which means that it must be affected. And that's why the connection between PNS and CNS is affected. So in my talk, I would like to say reactive astrogliosis. So astrocyte is a very common neuronal cell. It is a glial cell. And when there is any abnormality in the brain, so it proliferates, which is known as reactive astrogliosis. And like this. And microglia, it is also a glial cell, and when there is an abnormality, microglia also proliferate, and they actually try to defense, including phagocytosis. And the most important phenomenon about microgliosis is you can see this is the normal resting microglia, but this is the active microglia, which is very large in volume and also have more process. So the purpose of this study is that we already found neurofilament accumulation in the CNS, but in this study I will try to find out the relation of neurofilament accumulation with reactive astrogliosis and microgliosis. So as you can see, this is DST homozygote and this is wild type. So you can see this is the brain, this is called reticular nucleus, you can see, and you can see like here is lots of neuron is abnormally accumulated, stained by neurofilament. And you can see in wild type there is no such type of accumulation. And as I said that if there is any abnormal condition in the brain, so astrocyte responds very quickly. So I tried to find out what is the condition of astrocyte, and you can see this is the reactive astrocyte, and you can see lots of astrocytes in here, while in wild type there is not too much astrocytes. The same thing is also found in vestibular nucleus, which is this part, and you can see lots of neurofilament is accumulated, and as well as lots of astrocytes, while in wild type there is no astrocytes. So next I try to find out is those reactive astrocytes are occurring surrounding those abnormally neurofilament accumulated region, and that's why I perform double staining with GFAP. GFAP is the astrocyte marker and the neurofilament as well. So we can see that as I by dotted line you can see there are no astrocytes in here, and you can see by this circle we can see lots of neurofilament accumulation is found. And you can see by this dotted line, most of the astrocytes are basically found in this region, which means that the region where the neurofilament accumulation is found is mainly astrocytes is occurring. As well as in the vestibular nucleus, you can see there are no astrocytes and there are lots of astrocytes, and neurofilament accumulation is occurring in here as well in the mark, we can easily see. So next I checked, because this is the universal thing, while astrocyte is activated, then microglia should be activated. So I checked about the microglia activation in the reticular as well as vestibular nucleus. So you can see in the wild type there are very small size, as I said, but in wild type, in homozygote mice, you can see very big cell volume in both vestibular nucleus and reticular nucleus, and I quantify the cell body size, which is higher, significantly higher in DST homozygote mouse. And in order to check the activated microglia, there is a specific marker for activated microglia called CD68. It can specifically stain the active microglia. So I perform in situ hybridization with CD68, and you can see indicated by red arrow lots of positive cells which are absent in wild type, as well as in the reticular nucleus, as well as in the vestibular nucleus, as indicated by red arrow. So in order to confirm that those CD68 in situ hybridization is actually coming from the microglia, I perform CD68 in situ and EBA1 immunohistochemistry double staining. So EBA1 is the microglia marker. It can mark every microglia, active or non-active, or CD68 can only mark the active microglia. So you can see there are very big double staining cell here, lots of us, but we didn't see any such type of very big staining in the wild type, as well as in the reticular nucleus and also in the vestibular nucleus. Lots of double positive cells which are big in size, as I mentioned earlier. So the summary is that abnormal neurofilament was found in the reticular nucleus and vestibular nucleus, and I want to mention, which is very important, that those two nuclei are involved in movement control. So it should have some effect. And glial activation, astrogliosis and microgliosis was found in those two neurofilament accumulated regions. And so it ultimately means that those two regions are the main background cause of this type of phenotype in DST homozygote mouse. So the research, future one I want to share about that, you know that about different type of neurodegeneration like Alzheimer's disease, Parkinson's disease. In all those neurodegeneration diseases, there are lots of extragliosis and microgliosis. So I will look forward to this type of neurodegeneration is occurring in our DS2 homozygote mouse, as well as the activated microglia have some phagocytic activity, because you know that when the microglia is activated, it tries to engulf the abnormal cells. So I am looking forward to investigate all of the possibilities, and thank you all for listening. Thank you.
Thank you, Mohammed Ibrahim, for the great talk on the studies in the mice. Now the session is open for the discussion.
Okay, so you said about the dystonia gene? Dystonia gene. So could you correlate the function of this gene with the actin and myosin in the muscle? How could you?
Actually, as if I look backward here, the DST Lucas, you can see here we insert the lag G in CH1 and CH2, and those are the CH1 and CH2 are the domain which can synthesize the actin binding protein.
Okay.
So it is highly localized or highly correlated with the actin binding protein. And as I said, the paper, I forgot to mention the name. It is actually named that actin binding protein is disrupted in this mouse. So your point is 100% accurate, that it affects the actin binding protein as actin binding filament involved in muscle contraction.
What's the medicine available right now for this type of disease?
Actually, as I said, that is a very rare disease, and unfortunately, Japan is affecting the most. So maybe all lab is occurring, but still there is not 100% curable medicine yet. And actually, the data is so poor that the data for human affected person have not yet found, or something like that. So it's not easy to say that which drug can affect, but it was found that, as I said, the basal ganglia, which is the middle part of the brain, if there are some surgery occurred in the basal ganglia, it feels some improvement, but not 100%.
Maybe the gene therapy or the protein therapy may work.
Maybe.
Has any further question for this? So if not, join me to thank him again. Next, let me invite Mr. Ghulam Sarovar, and from now onwards it's an oral talk. We have 4 more oral talk in this session.
Assalamu alaikum wa rahmatullahi wa barakatuh. So me, Mohammed Ghulam Sarovar, I am actually now working on fish population genetics, and especially on mud crab population in Bangladesh. So I am working as a JSPS invited research fellow, and I did my PhD in the same department in 2004, and definitely on the fish genetics. So the title of my talk is the nuclear and cytoplasmic DNA molecular marker that reveal the fine-scale genetic structure of marine organism. And just I'm going to very the generalized way to present my work. And the first the figure show the inheritance pattern of this gene. The 2 types of the DNA. One DNA, that's come from the nucleus, resided as a chromosome. It's called the nuclear DNA, and another DNA that is resided outside the nuclear. It's called the cytoplasmic DNA or non-nuclear DNA also. So nuclear DNA normally comes from both parents, from father and mother, but the mitochondrial DNA, in case of animal, but for case of plant, they have another extranuclear DNA like from chloroplast. but the mitochondrial DNA comes only from the mother for inheritance pattern. So these two types of DNA actually play a role in lineage marker. Lineage marker actually passed down from generation to generation without having any change, especially some exceptional cases for mutation. And they can help to determine the family tree of any individual. At the X chromosome, normally they determine the parental lineage, and mitochondrial determine the maternal lineage. So there are some basic differences between this nuclear DNA and mitochondrial DNA. That's we call the cytoplasmic DNA also. The main difference is in their size. The size in nuclear DNA is around in the million, whereas the mitochondrial DNA in the 1,000. And they are in the shape. The nuclear DNA is linear, but mitochondrial is the circular one. And another, striking difference between, this DNA is the recombination. Nuclear DNA recombination actually is happened through the crossover during the meiosis, but no recombination actually happened in the mitochondrial DNA. And nuclear DNA has the less, the mutation rate because of it has high, the ability, the repair ability. If we compare with the mitochondrial DNA, because it has high mutational rate. So this is the typical sketch of mitochondrial DNA, and as a mitochondrial DNA, actually it has two regions. One is the coding region, another is non-coding. And there are three types of gene, in the mitochondrial DNA. One is just translational RNA, and another is ribosomal RNA, and another type of gene that is just used, in cellular energy production. This type of, the gene, and this 12S DRN is highly conserved. This region. This is, the highly conserved one. Even if you find the similarity or identity from the bacteria until the human being. So this type of gene actually used for study of phylogenetic study of higher categorical levels such as phylum and subphylum. And 16S rDNA, 16S the rDNA normally is also used for phylogenetic the analysis, but at the mid categorical level, such as the families or gene. But the protein coding gene, it's around 37 genes. These protein coding genes, they evolve very fast. So that's why this portion actually used for study of phylogenetic study at the very lower categorical, such as in just species at general level. So what about the status or fact of nuclear DNA? But this is the example for for a human cell, and we know in the human being or human cell they have 23 pair of chromosomes, and 99.9% is the identical between all these the DNA, and only 0.1%, it is around 3 million base account for variation. So this variation is actually interested for population or evolutionary geneticists. They just pay attention on this variation. They don't like even the similarity. And the control length of DNA from a single human gene, a single human cell is around 2 meters. Just only single cell, the control length of DNA. If you extract all the 23 or 46, total 46 chromosome, and just untwist and make it straight, then it's become just 2 meters long. But one adult individual, they have, it contains 3.7 to 10 to the power 13 cell, in one body. So if you multiply with this cell, even with the 2 meter, I mistake it, it's around 7.42 actually, 7.42, so this kilometer. And what about the distance between the sun and the earth? Because the distance between sun and earth is 1.10 to the power 8, whereas we have the total chromosome, the length is around, the 500 times greater than the distance of the sun and the earth. Subhanallah. Because we contact, because the long, it's a so long distance is resided in our body. If you, and it's not the, you know, the theoretical, it's either the physical one. Because you can measure it physically, not the theoretical, not the, you know, the hypothetical one. The real one, and physically you can determine this distance. Okay. So there's another interesting thing, the size, the genome size, and we find that the genome size for a human being is around 3.2 billion, and its content is around 25,000 genes, and has the chromosome number 46 in total, 23rd pair. And the average gene density is very, very less. But whereas for virus, the size of this, the genome size is very low. It's 0.292 million, but it has only 184 the gene, and that gene density is better, you know, the high. But the interesting thing for the one marine organism, or if you call also the aquatic animal, is the puffer fish. The size of genome is also in the million, the 390 million. But it has the same number of genes like the human. So the gene density is very high actually in the this fish. So the exceptional case, because if you find, you know, the phylogenetic tree, then you can find the aquatic organism in the middle actually. But this is the exceptional case that the number of chromosomes is also very high, as well as the genome size is very low, but the gene density is also very high, even also compared with the virus also. So this species actually become very popular now as a lab specimen for studying the functional of the gene. And we also, I told before that the geneticists, they have pay attention on the variability of the the nucleotide sequence. So how come this variation come from? The variation actually occurred by the mutation and sexual reproduction during the in meiosis level through crossing over. So if we find just one single mutation, it's called the single nucleotide polymorphism. Single mutation, it really doesn't tell very much thing, but if you extend, okay, such as more than, if you consider just 3 mutation point, one short stretch of DNA, and this is the possible combination. If you consider the 3 mutation, the possible combination is the 8. So each of the combination of each name is called the haplotype. So population geneticists, normally they identify the haplotype from the different population, and they just analyze the pattern of the population. The another marker is internal transcribed spacer, ITS. And this ITS, this size of the ITS gene is very specific, species specific actually. It's vary with the species. So one can easily distinguish the species, what is actually the overlapping with the other species. So in our case, in Bangladesh, we have the mud crab, but mistakenly, actually, we report this mud crab, the Psilocerata, but actually it's not the Psilocerata, because in my current study, we also settled down this issue that this species is the Psiloolivacea, because the base pair of the ITS gene is different among these, all these species, because they share the plasticity in their morphometric characteristics. Is it going to finish?
Yes.
Okay, this another technique is microsatellite. I'm not going to detail about the microsatellite. Normally, we do the data, all this data we input in this Harlequin software, and we analyze this. The software, and software just give some the indication or index. For marine environment, actually, it is hypothetically found that the maybe population will be the population is the panmectic, because of the distribution of the gene flow is very, very rapid. But In some cases, it's also found the cryptic population. It's very specific, and this is the isolated actually, this one. And if we find the haplotype distribution like this way, then we can easily call this population as a cryptic